So in this video we will going to discuss about the concept of gene gun. So this is another important technique for transforming any kind of plant cell or animal cell. Okay, so this method holds the same principle as the normal gun. Okay, so here we are using a pulse of helium as a pulling force. So in the cartridge we have micro projectiles which contain gold and tungsten coated with recombinant DNA. Okay, so now with the help of that pulling force, that is pulse of helium, a tremendous amount of speed is generated. Okay, so this tremendous amount of force will help us to target those micro projectiles towards the cell of interest. Okay, so as here we are using a too much amount of speed, so there should be a disc present or any kind of stop present. The stop will retard the speed of the micro projectiles. Okay, otherwise the micro projectiles will not only penetrate the cell by this way, but it also again penetrate the cell in this way. Okay, and as a result the micro projectiles will be lost. So there should be a disc stopped by the screen present that will retard the speed of the micro projectiles, that so that the micro projectile will only penetrate the cell for one time. Okay, and gets entrapped inside the cell. Okay, now inside the cell, in the reducing environment, the gold and tungsten will be separated from the gene of interest, and as gold and tungsten are not too much harmful for us, we can use them for the transfer of any kind of gene of interest in any kind of cells. Okay, so that is the concept of gene gun method. So here I want to discuss with you the most important thing. Despite the picture says that they have generated a plant protoplasm, but it is not required, because the tremendous amount of force that we are using give the micro projectiles enough ability to penetrate any kind of cell. Okay, that means we don't need to bother about what is the architecture of the cell. The if we will use the gene gun, the gene gun will allow the micro projectile to simply penetrate the cell. Okay, so that means cell culture will no longer be a problem for gene gun method. Okay, and another important thing, gene gun method will help you to place DNA and RNA exactly in which cell you need. Okay, that is why the use of viral vectors and various kind of toxic chemical treatment are simply cancelled after the development of the gene delivery, gene gun method as a gene delivery. Okay, so it is mainly useful for transformation of those plants, or it is mainly useful for transformation of those plants who shows less efficiency towards agrobacterium mediated gene delivery method, for chicken pea, sorghum, along with pigeon pea, rice, corn. Okay, so that is the two kind of uses. This method commonly employed for genetic transformation of plant and many organisms, and mainly for those plants who do not capable to show sufficient response to agrobacterium tumefaciens mediated gene transfer in rice, corn, wheat, chickpea, sorghum and pigeon pea. Okay, and we are also using gene gun method for those plant cells who have a less regeneration capacity. Okay, for that means two important application, that is transformation of those plant cell who have less regeneration capacity, and another important thing, transformation of those plant cell who show less response towards agrobacterium mediated gene delivery. Okay, especially in case of rice, corn, wheat, chickpea, sorghum and pigeon pea. Okay, so now important thing that I want to mention, that Advantages part I have told you. That simple convenient method involved coating DNA and RNA. And most important advantage, you don't need to bother about no longer about the architecture of the cell. It will ensure the entrapment of the DNA by the high level or tremendous level of force that we are using to target those microprojectiles towards the cell. Okay, it will eliminates the use of potentially harmful virus as a gene delivery system. Okay, so transformation efficiency is obviously lower than the agrobacterium mediated transfer, and multiple insertion of the same copy can causes the chance of gene silencing. Okay, random integration is also a problem, and target tissue should have some at least regeneration capacity. Okay, that is important, and as specialized equipment is needed, so I think I don't have to mention that the technique is too much costly, right? So that is the concept of our gene gun method. So let me revise again. So we are here using a pulling force. We are using pulse of helium as a pulling force, and in the cartridge we have microprojectiles who are coated with DNA molecules. Okay, and just like a gun, the microprojectiles will be targeted towards cell at a tremendous amount of force. Okay, and that is why for preventing the loss of the cell, we are using a disc stopped by the screen. Okay, that will retard the speed of the microprojectile so that they can only penetrate those kind of microprojectile once. Okay, and become entrapped inside the cell. Okay, so now the uses we are using for transformation of those plant who show less sensitivity towards Acarobacterium mediated gene delivery, for example rice, sorghum, chickpea, etc. Okay, and we are also using this method for transferring those plant cells who have less regeneration capacity. Okay, as well as transgene transfer toward any animal and any plant cell is necessary with the help of this method. Okay, advantages. So gene delivery by gene gun eliminates the use of various kind of harmful chemical use and various kind of the thing that we are using, for example various kind of viral vector we are used which may be harmful for the organism itself. So gene delivery method by using gene gun eliminates the need of using those kind of procedures. Okay, so disadvantages: transformation efficiency is lower than Agrobacterium. Okay, and if multiple insertion of the gene of interest occur, then there can be a possibility of silencing, gene silencing. Okay, and random insertion is also a problem, and obviously the process is very costly. Okay, so that is the concept of the gene gun method. So I hope this video will be helpful to you, and thank you for listening to this class.